Welcome back to The Explainer. Today, we're setting the stage for a truly fascinating journey. We're going to follow a single breath, tracing its path from the hidden electrical signals firing deep within your brain all the way down to the microscopic chemical reactions happening inside your lungs. It's this seamless, split-second cascade of biology and physics, and honestly, by the end of our time together, you'll probably never look at a simple breath the same way again.
So, let me ask you something. How many breaths do you take without even thinking about it? Seriously, try to consciously think, right now, about something your body does automatically thousands of times a day. Respiration operates almost entirely behind the scenes of our conscious mind. While you're sleeping, working, or just sitting there, an incredibly complex network is actively keeping you alive, maintaining the rhythm of your life without requiring a single thought from you.
Which brings us to our first key concept, eupnea. This is your quiet, restful breathing. This basic, restful rhythm is literally the foundational tempo of life. Maintained completely subconsciously, eupnea really only requires the activity of the dorsal group in your brain to activate your diaphragm and external intercostal muscles. Exhalation during eupnea? That's entirely passive. It just relies on the natural elastic recoil of your lungs. But where exactly is this foundational tempo being controlled?
Let's dive into Section 1, the brain's control room, specifically the medulla and pons.
To answer where that tempo is controlled, we've got to travel up into the brainstem. If we cleanly break down the dense anatomical geography here, we essentially have two major regions dividing the labor of breathing, the medulla and the pons. Down in the medulla, we have the dorsal respiratory group. Think of this as your pacemaker. It sets that basic rhythm we just talked about by taking in sensory info from peripheral chemoreceptors and stretch receptors in the lungs. Right next door is the ventral respiratory group. Now, this group is completely inactive during that quiet eupnea breathing. But when you need to breathe forcefully, like say during a heavy workout, this area kicks into gear to drive active expiration. Then moving up to the pons, we find the pontine respiratory group, which houses the pneumotaxic and apneustic centers. Their main job? Controlling the rate and the overall pattern of your breathing.
Now there's this absolutely beautiful neurological push and pull dynamic happening right there in the pons. You've got the apneustic center, which is constantly sending positive, stimulating impulses down to the medulla. It desperately wants to promote and prolong inhalation. But if it were just left unchecked, your breathing would be totally abnormal and sustained. So it's countered by the pneumotaxic center. The pneumotaxic center cyclically inhibits inhalation. It acts as the ultimate inspiratory off switch. It literally limits the burst of action potentials traveling down the phrenic nerve, which effectively regulates your tidal volume, the amount of air you actually take in, and keeps your respiratory rate nice and steady. It's just a perfect neurological balancing act.
Moving on to Section 2, the physics of the lungs, or what I like to call the engine room.
Let's see how this all translates into the physical hardware of our bodies as that electrical signal travels down to where mechanical expansion actually happens. Deep inside your lungs are the alveoli. You can kind of think of them as millions of tiny wet bubbles, spherical outcroppings surrounding a central airspace. And because they're wet, they behave a lot like gas trapped in water. This introduces a massive physical problem dictated by the Young-Laplace equation. Because of the surface tension of the water lining these tiny spheres, the alveoli naturally want to decrease their surface area. Basically, they inherently want to collapse in on themselves.
And here's the crucial number, 70 millinewtons per meter. That is the surface tension of normal water at room temperature. If the fluid lining your alveoli was just pure water, the collapsing force would be so incredibly strong that inflating your lungs with every single breath would be an impossibly exhausting feat. I mean, your muscles would fatigue almost immediately just trying to pull those microscopic balloons open against that 70 millinewton force.
Which brings us perfectly to Section 3, pulmonary surfactant, our biological soap. Enter the absolute hero of our lungs engine room. Pulmonary surfactant is this miraculous chemical complex formed by specialized cells called type 2 alveolar cells, and it prevents the engine of our lungs from seizing up. Contrast that 70 millinewtons per meter of normal water with this, less than 1 millinewton per meter. When you exhale and your lungs drop below 40% of their total capacity, the surfactant phospholipid molecules compress together. This compression drops the surface tension inside your alveoli to near zero levels. This is what allows your lungs to inflate easily on the very next breath, massively increasing pulmonary compliance and preventing a super dangerous condition called atelectasis, which is the complete collapse of the alveoli.
So what exactly is this miracle substance made of? Breaking down the chemistry, it's about 40% DPPC, which is a specific phospholipid with two saturated carbon chains. DPPC is without a doubt the strongest surfactant molecule in the mix. But here's the catch. On its own, its phase transition temperature is 41.5 degrees Celsius. That's much higher than our normal human body temperature of 37 degrees. This means pure DPPC would just be a slow, rigid gel sitting in our lungs. That's exactly why it completely relies on the other ingredients, the 40% other phospholipids, 10% neutral lipids like cholesterol, and 10% proteins. All these other elements fluidize the mixture, allowing the DPPC to spread rapidly across the air-water interface of the alveoli at our normal body temp.
Now, let's look a bit closer at that small but literally life-saving 10% slice, the surfactant proteins. They work synergistically to do two pretty amazing things. First, you have the highly hydrophobic proteins SBB and SBC. They grab onto the DPPC and massively increase the rate at which it spreads over the alveolar surface. Without them, our lungs would just fail biophysically. Second, proteins SBA and SBD act as a frontline defense for our innate immune system. They're collectins that bind to sugars on the surface of invading bacteria and viruses, opsonizing them, which is really just a fancy way of saying they coat the pathogens to make them highly visible and tasty targets for our immune system's macrophages to hunt down and destroy.
You also really have to marvel at the sheer recycling efficiency of the human body here. Surfactant was secreted by the type II pneumocytes and stored in these concentric rings called lamellar bodies. Once it's released into the alveoli, it forms a meshwork and functions efficiently for about 5 to 10 hours. But here's the incredible part. Up to 90% of the DPPC in the surfactant isn't just wasted. It's actively pulled back into the cell via a process called clathrin-dependent endocytosis, spurred on by that SPA protein. Your lungs are constantly reusing, refreshing, and literally sweeping up their own biological soap.
Let's shift gears for Section 4. When the system fails, infant respiratory distress syndrome.
Because we have to ask, what happens when this delicate chemical balance isn't fully developed? When you anchor this biology to clinical reality, the figures in human development are just staggering. Surfactant production only really begins in earnest around 20 weeks of gestation. A full-term infant is born with a robust alveolar storage pool of approximately 100 milligrams per kilogram of surfactant. But a preterm infant, born before 28 to 32 weeks, might only have an estimated 4 to 5 milligrams per kilogram. Without that biological soap, that 70 millinewton surface tension of water just takes over. The alveoli collapse, compliance plummets, and the infant simply cannot overcome the immense physical work required to breathe. This is what's known as infant respiratory distress syndrome, or IRDS.
Fortunately, medical science has completely stepped up to this immense challenge. To treat IRDS, we've developed artificial pulmonary surfactants, and they are so absolutely vital that they're formally recognized on the World Health Organization's model list of essential medicines. We have synthetic options like Exosurf and Lucinactin, which mix DPPC with specific spreading agents or synthetic peptides that mimic those crucial SP proteins. And then there are incredibly effective animal-derived options like Barictant, extracted from cows, and Poricant alpha, extracted from pigs. These medicines are administered directly into the lungs of premature babies, instantly dropping the surface tension and saving countless lives by perfectly mimicking the very system we've just been exploring.
So, as we wrap up this explainer, I want you to take a deep breath right now. Expand your chest. Let the air out. Can you feel it? In that single second, electrical signals fired from the dorsal group in your medulla, checked and balanced by the pneumataxic center in your pons. That signal shot down to your diaphragm. And as your lungs expanded, a liquid mixture of DPPC and specialized proteins rapidly spread across millions of microscopic bubbles, driving the surface tension down to near zero so that you wouldn't exhaust yourself just trying to stay alive. The dual-layered complexity of the neurological software and the biological hardware working together inside you is nothing short of an absolute marvel.